So dear students, as you know already, concerned with second chapter of biotechnology, initially we have come across three options to increase the food production. Agrochemical based agriculture, second is organic agriculture, third is genetically modified crop based agriculture. Followed by that we have come across a very interesting matter, applications of biotechnology in the field of medicine, where you have come across insulin production, very important for the point of examination. Followed by that you have come across gene therapy, molecular diagnosis and transgenic animal. An animal with a transgene are called as transgenic animal, or an animal with a genome that has been altered due to inclusion of foreign genetic material or foreign DNA is called as transgenic animal. 5 uses you have come across. Followed by that, now look over the last matter concerned with biotechnology. So today we are going to study ethical issues, patent and biopiracy, but it is the last matter. But don't omit the matter, because when you go through the previous theory papers, many times they have arisen the question, even few CT questions also they have prepared based on this information. Ethical issues, patent and biopiracy. Genetic engineering is commonly called as hot cake of today's high-tech world. Genetic engineering is commonly called as hot cake of today's high-tech world because it has many advantages and disadvantages, or it has its own positive and negative impact not only on the environment but even on human beings also. Genetic engineering is commonly called as hot cake of today's high-tech world because it has advantages and disadvantages, or you can call it as a merits and demerits, or it has its own positive and negative impact not only on the environment but also even on human beings. When it is utilized in a proper way, no doubt it has many benefits. When biotech and genetic engineering is used in a proper way, no doubt it has many benefits or advantages. But when it is not utilized in a proper way, or when it is misused, no doubt it will become hazardous enemy on humankind. When genetic engineering and biotechnology is used in a proper way, it has many advantages. But when it is not utilized in a proper way, or when it is misused, no doubt it will become hazardous enemy to humankind. Therefore, Government of India has made genetic engineering approval committee of Ministry of Environment and Forest. Therefore, Government of India has made genetic engineering approval committee of Ministry of Environment and Forest. It will take into account of GM research. It will take care of validity of GM research. It will take care of validity of GM research. Introducing genetically modified organism into the environment. Introducing genetically modified organism into the environment. Import and export, or import and export of genetically altered organisms, and even it will approve large scale manufacture. Look over here, dear students. Genetic engineering and biotechnology is having both merits and demerits. When it is utilized in a proper way, it has many advantages. When it is misused, no doubt it will become hazardous enemy to mankind. Therefore, Government of India has made Genetic Engineering Approval Committee of Ministry of Environment and Forest. It will take care regarding the validity of GM research, genetically modified organism research, validity of GM research, import and export of genetically altered organisms, introducing genetically modified organism into the nature or environment, approve large scale manufacture, Now look over, dear students, next matter is patent. It is an exclusive right granted by the government body either to an inventor or to an applicant over a period of 17 to 20 years that will prevent others from making, using and selling the invention. Look over here, you have to take care of this matter for 1 mark. Patent is an exclusive right granted by the government bodies either to an inventor or to an applicant over a period of 17 to 20 years that will prevent other from making, using and selling the invention. But many peoples are against this patent concerned with the companies. What they are using genetic material, plants and bioresources are actually identified, developed and used by the indigenous peoples and farmers of specific region. Look over the matter. Patent is an exclusive right granted by the government body either to an inventor or to an applicant over a period of 17 to 20 years that will prevent other from making, using and selling the invention. But many peoples are against the patent of the companies. What the companies are using? Genetic materials, plants, animals or bioresources are actually identified, developed and used by the farmers and indigenous people of specific region or it may be an country. I'll provide you one such an example, rice. You know already, dear students, there are many varieties of rice are there in India. Among them, the popular one is basmati rice. There are many varieties of rice are there in India. Among them, one is basmati rice. It is known due to its aroma and flavor. It is popular due to its smell and taste. There are about 27 documented varieties of rice are grown since from centuries. There are about 27 documented varieties of basmati rice are grown since from centuries. Even when you go through the ancient text and poetry, there also they have mentioned the matter related to basmati rice. Recently, in the year 1997, an American company, in the year 1997, an American company has got US patent. An American company has got US patent that has allowed the company to market new variety of basmati rice not only in the US and even abroad also. But actually they have produced this new basmati rice by crossing Indian farmer variety with that of semi-dwarf varieties. They are claiming that it is a new invention. Look over, dear students, and take care of this matter. Patent is an exclusive right granted by the government bodies either to an inventor or to an applicant over a period of 17 to 20 years that will prevent others from making, using and selling the invention. And many peoples are against the patent given to an companies because what they are using genetic material, plants, animals or bioresources are actually identified, developed and used by the farmers and indigenous people of specific regions. Okay, one such an example is rice. There are many varieties of rice are there in India. Among them, the popular one is basmati rice. It is known due to its flavor and aroma. There are about 27 documented varieties of basmati rice are there in India. They are grown since from centuries. When you go through ancient text and poetry, there also they have mentioned the matter related to basmati rice. In the year 1997, an American company has got US patent related to new basmati rice. That patent has given permission for this company to market basmati rice not only in the United States but also even abroad. But actually this new basmati rice they are produced by crossing indigenous or India's basmati variety with that of semi-dwarf variety. They are claiming that it is a new invention. Even many attempts are going on to take the patent. Many attempts we have seen concerned with the taking indigenous medicinal herbs or India's medicinal herbs like neem and turmeric. Therefore, being as an young generation, you have to take care of such issues. Otherwise, we are going to lose rich legacies. So, dear students, you please remember many attempts have taken already to take the patent concerned with the Indian medicinal plants like neem and turmeric. We have to take care of such issues. Otherwise, we are going to lose rich legacies. Now look at the last term, biopiracy. It is the use of bioresources. It is the use of bioresources by the multinational companies and other organizations without proper authorization from the country nor people concerned without adequate compensation. Biopiracy is the use of bioresources by the multinational companies and other organizations without proper authorization from the country nor people concerned without adequate compensation. ಸೊ ಡಿಯರ್ ಸ್ಟೂಡೆಂಟ್ಸ್, ನಿಮ್ಗೆ ಕೆಲವೊಂದು ಸಲ ಗೊತ್ತಿದೆ, ಕೆಲವೊಂದು ಫಿಲ್ಮ್ಸ್ಗಳನ್ನು ರೆಡಿ ಮಾಡಿರ್ತಾರೆ ಪ್ರೊಡ್ಯೂಸರ್ಸ್ ಮತ್ತು ಡೈರೆಕ್ಟರ್ ಕೊಟ್ಬಿಟ್ಟು. ಆದರೆ ಅದು ಇನ್ನೂ ಮಾರ್ಕೆಟಿಗೆ ರಿಲೀಸ್ ಆಗೋದಕ್ಕಿಂತ ಮೊದಲೇ ಯಾರೊಬ್ಬರು ಕಾಪಿ ಮಾಡಿ ಮಾರ್ಕೆಟಿಗೆ ಬಿಟ್ಬಿಡ್ತಾರೆ. ಹಂಗೆ ಬಿಟ್ಟಾಗ ಏನಾಗುತ್ತೆ, ಪ್ರೊಡ್ಯೂಸರ್ಗಳಿಗೆ ಮತ್ತು ಉಳಿದವ್ರಿಗೆಲ್ಲರಿಗೂ ಗ್ರೇಟ್ ಲಾಸ್ ಆಗಿ ಬಿಡುತ್ತೆ. ಅದನ್ನು ಅವಾಯ್ಡ್ ಮಾಡೋಕ್ಕೆ ಬೇರೆ ಬೇರೆ ಟೆಕ್ನಾಲಜಿಗಳುಂಟು, ಅದನ್ನೇ ನಾನು ಹೇಳ್ತಾ ಇರೋದಿಲ್ಲಿ. ಬಯೋಪೆರಸಿ ಈಸ್ ದಿ ಯೂಸ್ ಆಫ್ ಬಯೋರಿಸೋರ್ಸಸ್ by the multinational companies and other organizations without proper authorization from the country nor individual without giving proper amount, without giving a adequate compensation. But you know already, so many forwarded areas or many multinational companies, countries are very rich economically or financially. They are very rich, but at the same time they are very poor with respect to traditional knowledge and biodiversity. Biodiversity means it is a collection of varieties of all forms of life. Biodiversity is a collection of varieties of all forms of Biodiversity is a collection of varieties of all forms of life. Industrialized countries are financially they are very rich, but at the same time they are very poor with respect to traditional knowledge and biodiversity. Biodiversity is a collection of varieties of all forms of life. Don't bother about this biodiversity. Detailed explanation is there in the later classes. Okay, but developing countries are financially they are poor, but they are very rich with respect to traditional knowledge and biodiversity. Now, this traditional knowledge is used to produce modern applications. Traditional knowledge is used to produce modern application that will help to save the time, efforts and expenditure during commercialization. But many peoples are against these systems because many developing countries have recently identified, the injustice and inadequate compensation. Therefore, the Indian Parliament has recently permitted second amendment bill concerned with the patent that will take care of such issues. Okay, dear students, as you know, after completion of biotechnology, so today I will switch over to some of the previous questions. Okay, number one, NEET 2020. NEET 2020. Now, question arises, why I am discussing the previous question? Just I would like to prove that. So, if you are well versed with the theory, you can easily score good marks in NEET examination. Okay, question number one, which has appeared in NEET 2020. Choose the correct pair from the following. Choose the correct pair from the following. Number one, polymerases. Break the DNA into fragments. You know, break the DNA into fragment is performed by REN. Restricted endonucleases are commonly called as molecular scissors because it will cut the DNA at the specific site having palindromic sequence. But polymerases will play a significant role in the formation of polymers, or you can call it as a nucleic acids. Polymerases will play a significant role in the formation of polymers, or you can call it as a nucleic acids, or DNA or RNA. Nucleages. Restriction endonucleages will come under a large class of enzymes known as nucleages. Restriction endonucleages will come under a large class of enzyme known as nucleages. They are of two groups, exonucleages and endonucleages. Exonucleages will remove nucleotides from the end of DNA, but restriction endonucleages will cut the DNA. So nucleages separate the two strands of DNA. This line is also wrong. Exonucleages make cuts at specific position within DNA. This line is also wrong because exonucleages will remove nucleotides from the end of DNA. Now look for the last one. You know, it is commonly called as molecular glue or molecular stitchers because they will connect any two contents of DNA. So ligases will join the two DNA molecule. They will join the two DNA molecule. Therefore, they are called as molecular stitchers. Okay, so that ends question number one. Now look at question number two. In gel electrophoresis, separated DNA fragments can be visualized with the help of, back to theory matter. You know already, when the DNA will come under the action of restricted endonucleases, when DNA will come under the action of restricted endonucleases, fragments of variable sizes are produced. These fragments are separated by a technique known as gel electrophoresis. DNA fragments can be seen only after staining with ethidium bromide or ethidium bromide and exposed to ultraviolet radiation. Look over here. When the DNA will come under the action of restricted endonucleases, fragments of variable sizes are produced. These fragments are separated by a technique known as gel electrophoresis. DNA fragments can be seen only after staining with ethidium bromide or ethidium bromide and exposed to ultraviolet radiation. Under ultraviolet radiation, they appear dark orange color bands. These bands are separated and utilized in the process of recombinant DNA technology. Based on this explanation, the correct answer is ethidium or ethidium bromide in ultraviolet radiation. One more matter they have given, acetocarmine. It is used to stain chromosomes. Look over, dear students. This acetocarmine is used to stain chromosomes. You know already, chromosomes are the nuclear component having specific organization, individuality, structure and function. So the correct answer is number 1. Now look over question number 3. Okay. BT cotton variety that was developed by the introduction of toxin gene of Bacillus thuringiensis is resistant to insect pest. Okay. Here look over, one more new word is there. Predators are generally larger than the prey. Predators are generally larger than the prey. Predators will attack, kill and feed on the prey. Predators will attack, kill and feed on the prey. I will provide one simple example for the prey and predator relationship. Predator example is cat, prey example is rat. Cat will attack, kill and feed on the prey. But sometimes predator will also become prey for other organisms. That is a different story. Look over here, BT cotton variety that was developed by introducing toxin gene of Bacillus thuringiensis is resistant to insect pest, but not to insect predators. Fungal disease, plant nematodes. You know already, nematodes are the common parasites over plants, animals and even human beings, where RNA interference I had discussed. Look over next question number 4. Identify the wrong statement with regard to restriction enzymes. Dear students, so in NEET 2020, many students are get confused with this question. Take care, very simple question. Identify the wrong statement with regard to restriction enzymes. Option number 1, they cut the strand of DNA at palindromic site. They cut the strand of DNA at palindromic site. This line is correct. You know already, DNA restriction endonucleases will cut the DNA at the specific site having palindromic sequence. They are useful in genetic engineering, as already I covered, REN and ligase. Without these two enzymes, you cannot conduct any experiment in genetic engineering. Molecular scissor, molecular knives. Without these two, molecular knives or molecular scissor and DNA ligase, without these two enzymes, you cannot conduct any experiment in genetic engineering. Each restriction enzyme functions by inspecting the length of DNA sequence. As you know already, REN will first recognize us, then it will cut. The region which is recognized by the REN is called as recognition site, and where it will cut is called as restriction site. Now look over option number 3 carefully. Sticky ends. You know already, sticky end is nothing but a single stranded DNA, where you can join another single stranded DNA having complementary base sequences. Sticky ends means single stranded DNA where you can join another single stranded DNA having complementary sequences. Sticky ends can be joined by using DNA ligase. This line is correct, but it is not with respect to restriction enzymes. Look over, dear students, and take care of this question. Most of the students are get confused. Actually, it is a very easy question. Identify the wrong statement with regard to REN. But this line, sticky ends can be joined by using DNA ligase is correct, but it is not the function of restriction enzymes. As a result, correct answer is number 3. The specific palindromic sequence, dear students, palindromic sequence means the sequence is same when you read from opposite side on the two strands of DNA. Palindromic sequence is same when you read from opposite side on the two strands of DNA. Under that I quoted ECOR1 is an REN that will cut. The palindromic sequence is GAATTC. 5 prime 3 prime GAATTC. Same way at the lower side GAATTC. The palindromic sequence is same when you read from opposite side on the two strands of DNA, which is recognized by ECOR1. It is an restriction endonuclease. So the answer is number 4. Okay, you have to go through even all the options carefully, then you have to take the correct answer. Same question has once appeared in 2019 KCET examination. Same matter or same question has appeared once in KCET 2019, but slight changes they are made that I will discuss later. Now look at question number 6, which has appeared in NEET 2020. Match the following columns and select the correct option. So column number 1 and this is column number 2. Column number 1, Bt cotton, adenosine deaminase deficiency, RNA interference, polymerase chain reaction. Bt cotton, we can easily match the matter. It is Bacillus thuringiensis. Bt cotton, Bacillus thuringiensis. Look at this side option. If you match only one, you can easily guess the answer. But you should not move in such a way. You have to take care of all the 4 options. But here, a very easy matter. If you match the first matter, Bt cotton, Bacillus thuringiensis, you will get the answer. But with respect to other questions, you have to match all the 4 options, then you have to select the answer. Bt cotton, Bacillus thuringiensis. Adenosine deaminase deficiency gene therapy. You know already the technique of replacing the faulty gene, by a normal functional gene is called as gene therapy. First clinical gene therapy was given to a 4-year-old girl suffering from severe combined immunodeficiency disease, which is caused due to deletion of adenosine deaminase. RNA interference is a cellular defense mechanism which is noticed in the eukaryotic organism. RNA interference, cellular defense mechanism which is noticed in all the eukaryotic organism. So while explaining molecular diagnosis, previously what we are following the technique will not help us to identify the disease in the initial period. Therefore, advanced technologies like recombinant DNA technology, polymerase chain reaction, enzyme-linked immunosorbent assay tests are used to identify the diseases in the initial period. Polymerase chain reaction will help us to detect the presence of HIV in a suspected AIDS patient. Based on that, the correct answer is number 4. Okay, so look over the matter carefully and the correct answer is number 4. Which of the following statement, last question, which of the following statement is not correct? The proinsulin has an extra peptide called C-peptide. Now back to insulin production. While explaining insulin production, I put forward one important matter. Before insulin, a proinsulin is produced. Before insulin, a proinsulin is produced. It is having alpha chain with 21 amino acids, beta chain with 30 amino acids, connecting chain with 33 amino acids. Connecting chain is removed, and finally A and B or alpha and beta chains are linked by the disulfide bond to get an functional insulin. Before insulin, a pro-insulin is produced. It is having alpha chain with 21 amino acids, beta chain with 30 amino acids, connecting chain with 33 amino acids. Connecting chain is removed, and finally alpha and beta chains are linked by the disulfide bond to get an functional insulin. The proinsulin has an extra peptide called C peptide. This line is correct. Genetically engineered insulin is produced in E. coli. It is also correct. As already I covered, E. coli are frequently used as an host cell in genetic engineering. In man, insulin is synthesized as a proinsulin. This line is also correct. But look over the last option, the functional insulin. The functional insulin has A and B chains linked together by hydrogen bond. Actually, instead of hydrogen, it should be disulfide bond. Then it is the correct option. But here they are given not correct. So you have to take care of the questions. Which of the following statement is not correct? The functional insulin has A and B chains linked together by hydrogen bond. You have to take care, it is disulfide bond. So hydrogen bond is the wrong explanation. That ends 2020 questions. So total 7 questions have appeared from the biotechnology. Around 5 questions have appeared in KCET 2020. Based on biotechnology, 5 questions have appeared from the KCET. So finally it is very clear that biotechnology is having more weightage, not only concerned with the theory but also even NEET and KCET. Look over question number 1, which has appeared in KCET 2020. From the given combinations of steps in PCR, from the given combinations of steps in PCR, identify the enzyme dependent step. You know, polymerase chain reaction involves 3 steps: denaturation, annealing and primer extension. Polymerase chain reaction involves 3 steps: denaturation, annealing and primer extension. Primer extension requires TAQ polymerase. Primer extension requires TAQ polymerase. In addition to that, deoxynucleoside triphosphate and Mg++ are required for the primer extension. Based on that, the correct answer is A, extension only. Primer extension only. So DNA cannot easily enter into the cell. Back to theory matter. So DNA cannot easily enter into the cell. Therefore, you have to make the host cell as an competent. DNA cannot easily pass through the cell membrane due to hydrophilic nature. As a result, you have to make the host cell as an competent. Under that, various techniques we have come across, including heat shock, micro-injection, gene gun or biolistic, electroporation, etc. So gene gun or biolistic, where the plant cell is bombarded with high velocity microparticles of gold or tungsten coated with DNA. In a gene gun or biolistic, plant cell is bombarded with high velocity microparticles of gold or tungsten coated with DNA. Look over the question. Biolistic method is suitable for the gene transfer into plant cells. Okay. biolistic method is suitable for the gene transfer into plant cell, but microinjection in animal cell, microinjection in animal cell. Like this, you have to take care of the matter. So now look over the question which is related to gel electrophoresis. So before switch over to the question, try to recollect the matter which I have come across in my class. When the DNA will come under the action of restricted endonucleases, fragments of variable sizes are produced. These fragments are separated by a technique known as gel electrophoresis. Agarose gel is having sieving properties. Smaller molecules will move rapidly and reaches further end, whereas larger molecules will move slowly. Here in this picture, they are given 4 wells. This and larger DNA fragments or bands. This and smaller DNA bands. Very simple. M is largest DNA bands, N is smallest DNA bands. So finally, with the help of this example, it is clear that you have to take care even all the figures what they are given in the new NCERT book. In RNA interference, this word we have come across in nematode resistance. Okay. In RNA interference, the double-stranded RNA, DS stands for double-stranded RNA molecule will prevent. You know already, a double-stranded RNA will bind to specific mRNA. It will bind to specific mRNA and prevent translation of mRNA. As you know already, in molecular basis of inheritance, and even while explaining my chapter biotech, also I covered central dogma of protein synthesis, DNA replication, DNA transcription, RNA translation, protein. Double-stranded RNA will bind to specific mRNA and prevent translation. So this matter you have come across in molecular diagnosis. For effective treatment of the disease, early diagnosis is necessary. In addition to that, you have to know pathophysiology, altered physiology of the host. Previously or formerly we are using serum and urine test, but advanced technologies like recombinant DNA technology, polymerase chain reaction, enzyme linked immunosorbent assay test will help to identify the disease in the initial period. Look over the matter. Nowadays, the early diagnosis of the bacterial or viral infection in human is possible by using polymerase chain reaction. It helps in the amplification of DNA. Polymerase chain reaction. Serum analyzer is an old technique. DNA sequencer, CT scan, you know MRI and CT scans are done to identify the presence of cancer in a suspected persons. Okay, answer is polymerase chain reaction. So now students, please remember this matter. 7 questions have appeared in NEET 2020. 5 questions have appeared in KCET 2020. So now look over KCET 2019 question. In cloning vectors, you know already, vectors are the carriers of desired DNA. Vectors are the carriers of desired DNA. Under vectors, we have come across many examples, including plasmids. Under plasmids, I quoted 2 examples. One is PUC-18 and another one is PBR322. PUC-18 and another example is PBR322. Antibiotic resistant gene. 2 examples I covered. One is ampicillin, tetracycline, kanamycin. They will act as an selectable marker. It helps in the selection of recombinant from the non-recombinant. Very easy question. In cloning vectors, antibiotic resistant gene are helpful for selection of recombinants from the non-recombinants or transformants from non-transformants. Okay. Now, you know already, so a student while extracting DNA from fungus requires dash enzyme to break open this cell wall. Back to first step of recombinant DNA technology, isolation of genetic material. Back to recombinant DNA technology, first step we have come across isolation of genetic material, DNA, or it may be an RNA, but generally they are given preference for the DNA. A student while extracting the DNA from the fungus requires enzyme to break open this cell wall. DNA is found in all the living organism. Okay, DNA is found in all the living organism except few viruses. But DNA is abundantly noticed inside the nucleus. So first you have to break this cell to isolate DNA. Cellulase and pectinase are used to break the plant cell. Cellulase and pectinase are used to break the plant cell. Lysozymes are used to break the bacterial cell, whereas chitinase is used to break the fungal cell. So the correct answer is chitinase. Okay, chitinase is used to break the fungal cell wall. Which of these is not an advantageous in genetically modified crops? This matter we have come across in second chapter of biotechnology. Okay, so you go through all the options carefully, you will get an idea. It will increase the efficiency of using minerals. It will not depend upon the chemical substances or pesticides. The substance which are used to kill pests are called as pesticide. It has increased the nutritional value of the food. Under that I quoted golden rice as an example. While explaining golden rice, I put forward one more point. It is rich in vitamin A. And in addition to that, please remember the two donors of golden rice are daffodil and Erwinia iridoviria bacterium. And look over the last point, it will increase just the post-harvest losses. Actually, it will decrease, okay, not an advantage. It will decrease the post-harvest losses. So, correct answer is D. This question we have come across in the second chapter of biotechnology. Some multinational companies have exploited the traditional knowledge of the indigenous people to produce commercially important bioproduct without their consent. This is an example for biopiracy. You know already, biopiracy is the use of bioresources. It is the use of bioresources by the multinational companies and other organizations without proper authorization from the country nor from the individual person, without giving adequate compensation. So based on that, the correct answer is biopiracy. Okay. DNA precipitation out of mixture of biomolecules can be achieved by treatment with, you know already, cyanogen bromide, ethidium bromide or ethidium bromide and chilled ethanol. 3 points you have to take care. Chilled ethanol, ethidium bromide, cyanogen bromide. Chilled ethanol is used to precipitate DNA. Cyanogen bromide is used to separate beta galactosidase insulin production. Okay? Whereas ethidium bromide is used in gel electrophoresis. So answer is chilled ethanol. This question is totally related to bioreactor. You know, dear students, bioreactor is a container or a vessel. Bioreactor is a container or a vessel which are used to culture the cells Bioreactor is a container or a vessel which are used to culture the cell in controlled environmental condition for the production of some commercially valuable product. Bioreactor is a container or a vessel which are used to culture the cells at a controlled environmental condition for the production of some commercially valuable product. Which one of the following equipments is essentially required for growing microbes on a large scale for industrial production of enzymes? No doubt the answer is bioreactor. This one we have come across in microbes in human welfare, biological oxygen demand. This word is no more we have come across in biotechnology. So the correct answer is bioreactor. You know, this question is related to BT cotton or generally BT crops. So the toxin which is produced by the BT is not toxic to it. The toxin which is produced by the BT is not toxic to it because it is produced in an inactive form called as protoxin. When the insect or larvae will feed on these BT crops, protoxin will enter into gut region under alkaline pH 12. Under alkaline pH 12, protoxin is converted into active toxin. It will bind to the epithelial cells of the midgut region and produces pores. As a result, cell will swell and finally it will rupture or finally it will lysis, and the ultimate result is death of insect. Now look over what triggers activation of protoxin to active BT toxin of Bacillus thuringiensis in bollworm. It is alkaline pH of the gut. Alkaline pH of the gut, please remember it is 12. Hydrogen ion concentration is 12. The last question of NEET 2019. So this is the question which is related to golden rice. As previously I covered, golden rice is so named because the grains are yellowish in color. Golden rice is so named because the grains are yellowish in color. But here you have to take care of the donor, it is daffodil. Which of the following is true for golden rice? It has yellow grains because of a gene introduced from a primitive variety of rice. This line is wrong. It is vitamin A enriched, this line is correct, with a gene from daffodil. It is rich in vitamin A, but the donor is daffodil. The rest is, it is pest resistant with a gene from BT. That is wrong. It is drought tolerant, developed using agrobacteria as an vector. So the correct answer is, it is rich in vitamin or vitamin A, but the donor is daffodil. So that ends 2 years question papers. NEET 2020, KCET 20, NEET 2019, KCET 19 I covered. Like this, even you have to go through all the previous question papers so that you can easily develop confidence over the multiple choice questions.